чувство кворума. Помимо конкурентного вытеснения, сказывается также влияние чувства кворума. Что говорят об этом наши коллеги?
Справка. Многие живые микроорганизмы принимают коллективные решения демократическим путем при помощи так называемого чувства кворума. Это чувство появляется у микробов, когда происходит увеличение скученности отдельных особей. При увеличении скученности совокупность особей превращается в организованный коллектив. Микробы превращаются в дружное микробное сообщество. Благодаря чувству кворума бактерии чрезвычайно быстро общаются друг с другом посредством сигнальных молекул. Бонни Басслер, молекулярный биолог из США, в 2009 году говорила, что бактерии разговаривают друг с другом, используя химический язык, который позволяет им координировать защиту и монтировать атаки. Эта находка имеет потрясающие последствия для медицины, для промышленности и для нашего понимания самих себя.
Making light. And why this is interesting is because in the past decade we have found that this is not just some anomaly of this ridiculous glow-in-the-dark bacterium that lives in the ocean. All bacteria have systems like this. So now what we understand is that all bacteria can talk to each other. They make chemical words, they recognize those words, and they turn on group behaviors that are only successful when all of the cells participate in unison. And so now we have a fancy name for this. We call it quorum sensing. They vote with these chemical votes, the vote gets counted, and then everybody responds to the vote. And what's important for today's talk is that we know that there are hundreds of behaviors that bacteria carry out in these collective fashions. But the one that's probably the most important to you is virulence. So it's not like a couple bacteria get in you and then they start secreting some toxins. You're enormous. That would have no effect on you. You're huge. But what they do, we now understand, is they get in you, they wait, they start growing, they count themselves with these little molecules, and they recognize when they have the right cell number that if all of the bacteria launch their virulence attack together, they're going to be successful at overcoming an enormous host. So bacteria always control pathogenicity with quorum sensing. And so that's how it works. We also then went to look at what are these molecules. So these were the red triangles on my slides before. And so this is the Vibrio fischeri molecule. This is the word that it talks with. And then we started to look at other bacteria, and these are just a smattering of the molecules that we've discovered. And what I hope you can see is that the molecules are related. So the left-hand part of the molecule is identical in every single species of bacteria. But the right-hand part of the molecule is a little bit different in every single species. And what that does is to confer exquisite species specificities to these languages. So each molecule fits into its partner receptor and no other. So these are private, secret conversations. These conversations are for intra-species communication. Each bacteria uses a particular molecule, that's its language, that allows it to count its own siblings. And so once we got that far, we thought we were starting to understand that bacteria have these social behaviors, but what we were really thinking about is that most of the time bacteria don't live by themselves. They live in incredible mixtures with hundreds or thousands of other species of bacteria. And that's depicted on this slide. This is your skin. So this is just a picture, a micrograph of your skin anywhere on your body. It looks pretty much like this. And what I hope you can see is that there's all kinds of bacteria there. And so we started to think, if this really is about communication in bacteria, and it's about counting your neighbors, it's not enough to be able to only talk within your species. There has to be a way to take a census of the rest of the bacteria in the population. So we went back to molecular biology and started studying different bacteria. And what we found now is that in fact bacteria are multilingual. So they all have a species-specific system. They have a molecule that says me. But then running in parallel to that is a second system that we've discovered that's generic. So they have a second enzyme that makes a second signal, and it has its own receptor, and this molecule is the trade language of bacteria. It's used by all different bacteria, and it's the language of interspecies communication. And so what happens is that bacteria are able to count how many of me and how many of you, and they take that information inside and they decide what tasks to carry out depending on who's in the minority and who's in the majority of any given population. And so then again, we turned to chemistry, and we figured out what this generic molecule is. So that was the pink ovals on my last slide. This is it. It's a very small 5-carbon molecule. And what the important thing is that we learned is that every bacterium has exactly the same enzyme and makes exactly the same molecule. So they're all using this molecule for interspecies communication. So this is the bacterial Esperanto. And so once we got that far, we've started to learn that bacteria can talk to each other with this chemical language. But what we started to think is that maybe there's something practical that we can do here as well. So I've told you that bacteria do have all these social behaviors that they communicate with these molecules. And of course, I've also told you that one of the important things they do is to initiate pathogenicity using quorum sensing. So we thought, what if we made these bacteria so they can't talk or they can't hear? Couldn't these be new kinds of antibiotics? And of course, you've just heard and you already know that we're running out of antibiotics. Bacteria are incredibly multi-drug resistant right now, and that's because all of the antibiotics that we use kill bacteria. So they either pop the bacterial membrane, they make the bacterium so it can't replicate its DNA. We kill bacteria with traditional antibiotics, and that selects for resistant mutants. And so now, of course, we have this global problem in infectious diseases. So we thought, well, what if we could sort of do behavior modifications? Just make these bacteria so they can't talk, they can't count, and they don't know to launch virulence. And so that's exactly what we've done, and we've sort of taken 2 strategies. The first one is we've targeted the intra-species communication system. So we've made molecules that look kind of like the real molecules, which you saw, but they're a little bit different. And so they lock into those receptors and they jam recognition of the real thing. And so by targeting the red system, what we are able to do is to make species-specific or disease-specific anti-quorum sensing molecules. We've also done the same thing with the pink system. We've taken that universal molecule and turned it around a little bit so that we've made antagonists of the interspecies communication system. And these, the hope is that these will be used as broad-spectrum antibiotics that work against all bacteria. And so we think that this is the next generation of antibiotics, and it's going to get us around, at least initially, this big problem of resistance. So what I hope you think is that bacteria can talk to each other. They use chemicals as their words. They have an incredibly complicated chemical lexicon that we're just now starting to learn about. And of course, what that allows bacteria to do is to be multicellular, right? And so in the spirit of TED, they are doing things together because it makes a difference, right? So what happens is that bacteria have these collective behaviors, and they can carry out tasks that they could never accomplish if they simply acted as individuals. And what I would hope that I could further argue to you is that this is the invention of multicellularity. Bacteria have been on the earth for billions of years. Humans, a couple 100,000. So we think bacteria made the rules for how multicellular organization works, and we think by studying bacteria, we're going to be able to have insight about multicellularity in the human body. So we know that the principles and the rules, if we can figure them out in these sort of primitive organisms, the hope is that they will be applied to other human diseases and human behaviors as well. I hope that what you've learned is that bacteria can distinguish self from other. So by using these 2 molecules, they can say me and they can say you. And again, of course, that's what we do both as in a molecular way and then also in an outward way. But I think about the molecular stuff. This is exactly what happens in your body. It's not like your heart cells and your kidney cells get all mixed up every day. And that's because there's all of this chemistry going on, these molecules that say who each of these groups of cells is and what their tasks should be. And so again, we think that bacteria invented that, and then you've just evolved a few more bells and whistles, but all of the ideas are in these simple systems that we can study. And then the final thing is, again, just to reiterate that there's this practical part, and so we've made these anti-quorum sensing molecules that are being developed as new kinds of therapeutics. But then to finish with a plug for all the good and miraculous bacteria that live on the earth, we've also made pro-quorum sensing molecules. So we've targeted those systems to make the molecules work better. And so remember, you have these 10 times or more bacterial cells in you or on you, keeping you healthy. What we're also trying to do is to beef up the conversation of the bacteria that live as mutualists with you in the hopes of making you more healthy, making those conversations better so bacteria can do things that we want them to do better than they would be on their own. And I want to thank you for inviting me here. It's a big treat for me to get to come to this conference.